Let's get started. Our speaker today is Dr. Gufei Zhao. He's the assistant professor in our department in the Division of Developmental Biology. So, Dr. Gufei got earned his PhD from University of Southern Alabama in 2004, and then he came to Chicago and joined Northwestern University as postdoctoral fellow and Division of Pulmonary and Critical Care Medicine. He worked with Dr. Yasser Snyder, Division Chief of Pulmonary at the Northwestern University. In 2009, he came to UIC. Dr. Raj recruited him as a junior faculty, and since then he's here, and he doing extremely well, and he got an R01, funded R01, and he has about 35 publications in peer-reviewed journals. And last year he was given a American Thoracic Society Rising Star of Research Award. And so with that, he's going to talk today is PhD in hypoxia-induced pulmonary hypertension.
Let me turn off the light. Well, thank you very much, Dr. Reddy, for the kind introduction. So, as Dr. Reddy, you mentioned, I've been here since early 2009. Can you hear me here better now? You can. Okay. Does anybody hear me okay in the back? Okay. All right. So I'm trying to raise my voice a little bit more. So today I'm going to talk about the study we did regarding pulmonary hydroxylase 2 in hypoxia-induced pulmonary hypertension. So this, we call it PhD2, but it's not a second generation of Doctor of Philosophy anyway. So I don't have anything to disclose. So I'm going to give you an outline of what I'm going to talk today. So first, I'm going to introduce hypoxia signaling in pulmonary hypertension. And then I will introduce to you about how does protein hydroxylase 2 play a role in pulmonary hypertension. And then I will also introduce some mechanism regarding the regulation of protein hydroxylase in pulmonary smooth muscle cells. So first, I'm going to talk to you about a little bit about hypoxia. What is hypoxia? What does hypoxia do to human body or human cells? So actually, everybody in here today have been breathing air to maintain the oxygen homeostasis. And actually, it's not just us, it's all mammalian cells require oxygen, not only for energy production, that's particularly what we usually say, but also oxygen is part of some protein function as well. And how do we supply enough oxygen? We breathe. That's how our lung does in our body and the circulation system. Now, what if we don't have functional breathing or circulating system? That's where breathing disorders will lead to inadequate supply of oxygen, and that's what we call hypoxemia or hypoxia. It depends on what system are you talking about. Now, unfortunately, hypoxia is a part of our life. Some are normal, some are abnormal. Now, for lung, particularly in lung diseases, hypoxia is everywhere, actually in all the kind of lung disease. I'm just giving you two examples of hypoxia in lung disease. One is because of the ventilation perfusion mismatch. For example, in asthma patients in pulmonary embolism patients or people with COPD or people with interstitial lung disease. They have abnormal lung function. That's why they got hypoxia. And with people with shunt disease, pneumonia, pulmonary edema, you also see hypoxia in their body or in their cells. So basically, hypoxia is a common phenomenon in our body, whether it's physiological or pathological. Now, mammalian cells develop a way to adapt to this kind of oxygen dysfunction regulation. So how does cell regulate oxygen homeostasis? So the key part is a molecule called hypoxia-inducible factor. This is a transcription factor can induce VEGF, TGF-beta, or induce erythropoietin, or induce vasodilation in vascular system, but in pulmonary vessel system, actually vasoconstriction, and also induce glycolysis. So mostly we focus on glycolysis because if you don't have oxygen, your energy production is low. The way the body to adapt to it is to push up the glycolysis to induce ATP generation in that system. Now, in all the other pathways, whether it's EPO induction or VEGF induction, that will help generate more red blood cells or more vessels. That will help to deliver oxygen to the body, to the cells. So that's how the hypoxia-inducible factor regulate the oxygen homeostasis. Now, hypoxia-inducible factor itself is regulated. That is regulated by protein hydroxylase, PhDs here. So in normal conditions, like we are now, PhD activity is inhibited. This inhibition will lead to the hypoxia-inducible factor stabilization and activation. So nothing's going to happen. Now, what if we have a low oxygen? The low oxygen level will be sensed by protein hydroxylase. This case, the protein hydroxylase activity will be inhibited. Then you will trigger the stabilization or activation of hypoxia-inducible factor, and you have all the ATP generation by glycolysis, VEGF, and angiogenesis. So this is how our body adapts to the low oxygen conditions. However, if the adaptation goes wrong, this same mechanism will induce certain genes and contribute to the pathophysiology. In this case, is a pulmonary hypertension. So we know that hypoxia-inducible factor can induce endothelin, which can induce vasoconstriction, can induce GLUT1 and other glycolytic enzymes, which will shift the oxidative phosphorylation to glycolysis, just like the cancer cell did. And they will also induce PDGF and VEGF to induce cell growth and cell proliferation, which will increase the thickening of the blood vessels. All these contributing to the pulmonary hypertension. Now, that's also one reason that when we classify pulmonary hypertension, the third category is all about pulmonary hypertension due to lung disease and or hypoxia. So in some disease status, in some of our studies, we take some samples from patients with pulmonary artery hypertension. Now, since we're in pediatrics, actually we do have a one category is called persistent pulmonary hypertension of the newborn, PPHN. That is specific for pediatric pulmonary hypertension. So no matter how complex the pulmonary hypertension classification is, they all share the common feature, the definition. So the definition of the pulmonary hypertension, no matter the pediatric, no matter it's adult, or no matter it's a pulmonary artery hypertension or hypoxia-induced pulmonary hypertension, it's very simple. Hypertension represents a mean pulmonary artery pressure greater than 25 millimeter mercury. So that is very simple, straightforward definition of pulmonary hypertension. Now, disregarding all kinds of different etiology, pulmonary hypertension all actually share the same pathological features. For example, they have all kinds of functional and structural changes in the pulmonary vasculature. This change can be because of sustained vasoconstriction or pulmonary vascular remodeling, which we're going to talk about later on. Or it's because of in situ thrombosis, or just because of increased vascular wall stiffness. This all cause the remodeling or structural change in the pulmonary vasculature. This will increase the pulmonary vascular resistance, and then will increase the pulmonary artery pressure, leads to the pulmonary artery hypertension. However, the patient with the pulmonary hypertension does not die from pulmonary hypertension. They actually die from right heart failure, because if you have a high blood pressure, the heart has to work harder to help deliver the blood to the pulmonary, and if the heart cannot take it, they're going to die. Now, we know a lot now about how does this structural remodeling happen, and this actually shows the multiple signaling pathways has been implicated in the pulmonary hypertension. So on the left side, you will see a normal simplified pattern of the artery vessel, and in the right, that's a diseased vessel. As you can see, in diseased status, you have increased layer of endothelial cell layer. You have a significant increase of smooth muscle cell layer here, and here you will have adventitial interstitial fibroblasts as well. Many signals have been contributing to that. Growth factors like TGF-beta, and the HIF pathways here, and also the nitric oxide pathways, the PKG pathway, all has been known to contributing to this part. And based on what we know, and many drugs to treat pulmonary hypertension is also targeting on these pathways. For example, the PKG part of the nitric oxide pathways, sildenafil, which regulated the PDE pathways. So we know a lot already about these pathways. However, the 5-year survival rate of people with pulmonary hypertension still remains about 57%. So this disease still is a lethal disease without a cure right now. So which would suggest that we need to know better about how this disease happened and what is contributing to that disease, and what can we do. If we can find new targets, we probably can develop a new methodology to treat this disease. So since we know there are many different cell types involved in this disease, and endothelial cells, smooth muscle cells, and adventitial fibroblast cells. So if you look at the previous picture, the most dramatic increase of thickening is in the smooth muscle cells. So that's why we focus on smooth muscle cell biology in this disease. So now, with that, so we want to know more about this disease, so that's what we're going to talk to you about. The second part of my talk is about the proline hydroxylase 2 in pulmonary hypertension. So we know what's happening in patient. We can have the samples, but we cannot really directly working on human bodies to tease out all the signal pathways in the system. So what we can do in the lab is taking cell culture or taking animals to try to mimic what's happening in the human patient. So one way we do in the lab to mimic pulmonary hypertension is culture cells in hypoxic conditions. That's also fits in hypoxia-induced pulmonary hypertension. So that's what we do. This is a hypoxia chamber we use in the lab. We can reduce the oxygen level as low as 1%. Typically, for smooth muscle cell culture, we use 1 to 3% of oxygen conditions. Now, how do we know that we reach this hypoxia condition in this system, in the cells? So we measure the PO2 in this culture condition. So we start with the normal air. It's about 165 millimeter mercury. So that's about 21% of ambient air. So after about 1 hour, you can see the cells will have a PO2 about 20 millimeter mercury. This is about 1 to 2% of oxygen. So that's what the condition we have been using. Now, what if we take the cells out? Do they retain the oxygen levels? So as you can see, after we take out the cell out of hypoxia chamber, the oxygen pressure comes back. So basically, this system can mimic hypoxia, and it's reversible. We can control normoxia, hypoxia, we can reoxygenate the cells. So that gives us the freedom to study how cells respond to hypoxia signalings. Now, oftentimes, the cell level does not really mimic what's happening in a body, because the human body contains so many different cell types, so complex structures. So we need to do some animal studies. So in animal studies, what we do is very simple. Take the mice or rats, put in hypoxia chamber like here. So in hypoxia chamber, we control the oxygen concentration to 10% of oxygen. In 3 to 5 weeks, typically what we see pulmonary hypertension in these animals. So this is a system we have in the animal facility here. So hypoxia chamber here, this is the liquid nitrogen. So we infuse liquid nitrogen to the chambers to lower the oxygen levels over there. And we have monitoring each chambers to make sure we have 10% of oxygen in our system. So now we have the tools. So I want to show you some control data to see this hypoxia condition is sufficient or is able to induce pulmonary hypertension. So this is an example of a mice hypoxia condition. We put the mice in 10% of oxygen for 4 weeks and measure the right ventricular pressure. This is the pressure tracing. In normoxia, in normal conditions, the pressure here is about 20 to 23 mercury, millimeter mercury. In hypoxia conditions, you got about a 30 millimeter mercury. So it is hypertension. And the ratio of right ventricular hypertrophy or the, we can see also have increased right ventricular hypertrophy here. And look at the vessel, normal lung vessel, you have this thickening of the vessels, but in hypoxia, you have a significant thickening of the vessels. And this is the quantification of the vessel thickening. Now, if you take a look at the vessel in the lung, what you see in normoxia, so this is an angiography of the lung of the animals in normoxia or hypoxia. So each branching is basically a vessel. So what you see here, the difference between normoxia and hypoxia group is one, you have in hypoxia group, you have less branching of the vessels. Two, you have less conjunctions. So basically it's also consistent with the less branchings. Three, each vessel is shorter. So basically in pulmonary hypertension, mice, you have abnormal vessels structure there. So now we have a tool. So next we want to find out a target. What are we going to study? So there are so many genes out there. Now, since we know hypoxia-inducible factor is controlling pulmonary hypertension, there are a lot of enzymes also regulating hypoxia-inducible factor. Can these genes also play a role in hypoxia-induced pulmonary hypertension? So that's why we're looking back to the proline hydroxylase PhDs. So PhD has 3 isoforms, PhD 1, 2, and 3. What they do is they give hydroxylation to the hypoxia-inducible factor alpha. This hydroxylation will lead to the recognition by a ubiquitinase E3 ligase, von Hippel-Lindau protein, and finally leads to the protein cell degradation. So basically, if hypoxia-inducible factor is driving pulmonary hypertension, if somehow we can overexpress PhDs, so this overexpression PhD will inhibit HIF, and therefore we will inhibit the pulmonary hypertension. So that's the whole concept of studies. So now, since PhD has 3 isoforms, so which one should we pick? So now, if we look at the enzyme activity in the cell, typically all isoforms are expressed. However, 95% of activity comes from PhD. So that's why we focus on the PhDs in this disease. So how do we do it? So basically we do a gain of function, loss of function approach. First, we want to overexpress it. In this case, we overexpress PhD2 or PhD1 in the pulmonary artery smooth muscle cells. So we want to make sure they are overexpressed. Here is overexpression of PhD2, and we use PhD1 as a control, and the PhD1 is also overexpressed. So our overexpression is working. Now we want to see whether this overexpression will inhibit HIF. HIF alpha subunit has two isoforms, 1 alpha and 2 alpha. They have some overlap functions, but they also have a distinct functions. So we want to differentiate these two isoforms as well. So when we look at HIF1 alpha, after we overexpress HIF PhD2 or PhD1, the HIF1 alpha level is down as we would expect. However, HIF2 alpha is not a change at all. So basically in this system, PhD2 and HIF1 alpha is the signal we are mostly looking at. Now, what's going to happen to the cells? So we look at the cell proliferation. We measure PCNA, which is a cell proliferation marker. So you see after overexpression PhD2 or 1, you see less PCNA. So these cells are growing less. Now, smooth muscle cells is a differentiated cell type. So we want also to see how does that affect the smooth muscle markers. So we look at SMA, SM22 alpha, calpone. They don't seem to change. So mostly from our data, overexpression PhD2 is affecting cell growth, but not cell differentiation. So in addition to measure cell growth, we also measure the cell DNA replication, which is another way of measuring cell proliferation. So as you can see, after we overexpress it, the cell growth, the DNA replication is going down in pneumoxia and also in hypoxic conditions. So basically, PhD2 overexpression can inhibit cell growth in both pneumoxia and hypoxia conditions. And meanwhile, the cell death is increased after you overexpress PhD2 in hypoxic condition, but not in pneumoxic condition. This case, we are measuring the LDH activity, lactate dehydrogenase. This is a measure of cell death. Data cells will release LDH to the media. So if you have more activity of LDH, you have more cell death. So basically, all the data suggests that the PhD2 overexpression will inhibit cell proliferation by increasing cell death. Now, next thing we want to see, what if we take out PhD2 out of the picture? What's going to happen? So in here, we use a small RNA interference to silence PhD2 or PhD3 or PhD1. So we basically silence all of them individually and in combination. So we do see the silencing is achieved. And then we look at HIF1 alpha and 2 alpha. As you can see here, silencing of PhD2 again increase HIF1 alpha level. PhD1 also increased HIF1 alpha, but not PhD3. In the HIF2 alpha, somehow PhD2 silencing does not have a role on HIF2 alpha, but PhD1 can affect the HIF2 alpha. So in this case, again, we are showing in smooth muscle cells, in pulmonary artery smooth muscle cells, PhD2 HIF1 alpha is very specific in this cell type. Now we want to see what's going to happen to the cells after we silence PhD2. Again, we measure the PCNA as a way of measuring cell proliferation. After we silence PhD2, we increase cell proliferation here. Not much change on SMA myocardium or SM22 alpha or PD-Lin5, which is another gene we measured before, or calpone. Basically, again, we are seeing that the PhD2 is controlling cell proliferation. So this is a quantification here. And this is another way of measuring cell proliferation by measuring cell viability. Silencing PhD2 increase cell viability. Here is the DNA replication measured by BRDU incorporation. So again, all these data by overexpressing PhD2 or silencing PhD2, we do see PhD2 can control cell proliferation through HIF1 alpha. Now, this has all been done in cell culture models. So next, we want to move into animal models. So in this case, we create a smooth muscle cell specific knockout of PhD2 mice. So in this case, we have a PhD2 floxed mice, and we have a SMMHEC driven Cree mice. So by crossing Cree to floxed mice, we are able to knock out PhD2 in this case. Now, this actually Cree is driven by the estrogen receptor. So basically, this is an inducible model. So we can control the knockout whenever we want, wherever we want. So this is the genotype of these mice. So this is wild type mice, and this is the knockout mice we want. This is wild type mice. This is heterozygous. So we want take these homozygous knockout mice to do experiment. Now, before we do the animal experiment, we want to make sure this system indeed knock out the PhD2 in smooth muscle cells. So we did this confirmation experiment. Basically, in this case, we inject tamoxifen to induce Cree activity to knock out PhD2. And we use corn oil injection as a control. And we isolate the pulmonary artery smooth muscle from these mice, and we look at the PhD2 levels. So wild type corn oil inject mice and still have PhD2 expression. And when you inject tamoxifen, you're able to knock out or knock down PhD2 here and here. It looks like it takes some time, takes a week or so to be able to see the knockout of PhD2. Now, this knockout is very specific because we don't see any change on PhD1, PhD3. So basically, this is a system we are confirming we truly have a smooth muscle cell specific knockout of PhD2 mice. Now we're ready to do experiment. So in this case, first we want to see whether this knockout will increase the development of pulmonary hypertension as we expect. For this purpose, we inject these mice with tamoxifen to induce knockout first, and then put them in hypoxia for 4 weeks. At the end of the study, we measure the phenotype of these mice. So the first measurement is the RVSP, the pressure measurement. Hypoxia in control mice does induce the pressure as we would expect pulmonary hypertension. However, knockout of PhD2 has nothing to do with this hypoxia-induced pressure change. We also measured the RV hypertrophy, no change from here to here. However, when we look at the vessels, when we knock out the PhD2, we have even more vessel thickening than control mice. So basically, the knockout of PhD2 is enhancing the hypoxia-induced pulmonary hypertension. This is a picture for the vessels in wild-type mice, normoxia mice. This thickness of the vessel, hypoxia, you have more vessel wall. And in normal conditions of the knockout mice, you have similar vessel structure, but in hypoxia mice, you have dramatic increase in vessels. So this is what we are trying to see if we take out a PhD out. So what's going to happen with hypoxia-induced pulmonary hypertension? Now, for the therapeutic view, it's better to see whether you can't do anything after you have onset of the pulmonary hypertension. So in the next experiment, what we want to see is what if you have the hypertension first, and then you take out the PhD2? Do you see even worse hypertension? So in this case, we expose mice to hypoxia for 2 weeks first. Typically, after 2 weeks exposure, you will see increased pressure. It's about 28 millimeter mercury, so it's a pulmonary hypertension. And then at that time, you already have hypertension, so you inject tamoxifen to knock out the PhD2. And you wait for another 2 weeks to measure phenotype of these mice. So first, we measure the pressure In this case, you have much more pressure than the wild type mice. So basically knock out PhD to push up the RVSP even further in hypoxia. No change on the RV hypertrophy, no change of wall thickness change. So again, this experiment suggests that if you take out the PhD2 out of the patient which already have pulmonary hypertension, their pressure will keep increasing, so make the phenotype of hypertension even worse. So all these data suggests that PhD2 is playing a role in development and the progress of the pulmonary hypertension. So with that, so we want to try to come up with some ideas whether we can targeting PhD2 to treat pulmonary hypertension. That comes in with the next experiment. In this case, we inject these mice with a chemical compound called R59949. This compound has been known to activate PhD2s in cancer cells. So we want to see if PhD2 is controlling HIF and hypertension. So if we can somehow activate PhD2, we should be able to lessen pulmonary hypertension. So again, for this experiment, we are more focusing on the therapeutic potential. So we induce hypertension first by putting these mice in hypoxia for 2 weeks, and then we inject this chemical compound by 5 days, 1 day, 1 injection, for 2 weeks injection, each once a day. And then after 28 days, we'll measure the phenotype of these mice. So we saw that injection of this compound did not change the RVSP or the RV hypertrophy, but we do see the decrease of the vessel thickening. This image is showing here. This is just the wild type mice, DMSO as a control for this compound. So hypoxia has induced vessel thickening, and after this compound treatment, this thickening is gone. So basically, this chemical can activate PhD2, although we are not able to see the full reversion of the pulmonary hypertension, but we do see the reversion of the vessel thickening. At least partially, we are able to reverse pulmonary hypertension. That suggests that PhD2 most likely can be used as a therapeutic target for later development of pulmonary hypertension. Now, the reason why I'm saying that is this compound has been shown as a PhD2, but it's activated, but it's not very specific. PhD2 is basically dioxygenase. So this compound can also inhibit or activate other dioxygenase, such as the collagen dioxygenase. So that is not a very good way for the development of a drug for this disease. But this will give us a hint, at least, that if we can somehow specifically activate PhD2, we might have a better inhibition in this case. So by now, hopefully I can convince you that PhD2 is playing a role in pulmonary hypertension by inhibiting HIF, and that will cause the inhibition of pulmonary smooth muscle cell proliferation, and at the end, the inhibition of pulmonary hypertension. So that's how we, so far, we are able to see here. Now, yes, so by now we can see the PhD2 is there in pulmonary hypertension. So is this clinical relevance? So we're gonna to go to the last part of my talk regarding the regulation of proline hydroxylase 2 in pulmonary hypertension. Now, first of all, we want to see whether PhD2 expression is changed in patients with pulmonary hypertension. So in this case, we are able to obtain pulmonary artery smooth muscle cells from normal donors or from patients with idiopathic pulmonary hypertension or the associated pulmonary artery hypertension patient. So we measure the PhD2, PhD3, and PhD1 expression. So here is the protein expression level. In IPH patient, we don't seem to to be able to see difference. But in patients with associated pulmonary arterial hypertension, we actually see decreased PHD1, no change on PHD2 and PHD3. Next, we look at the mRNA levels of PHD1, 2, 3. So in mRNA levels in patients with idiopathic pulmonary hypertension or associated pulmonary hypertension, PHD1 mRNA is down, but the PHD2 mRNA is up. So these data suggest, yes, in patients, the PHD1 and the PHD2 protein level or mRNA level are changed. That shows the clinical relevance of the PHD2 or 1 in pulmonary hypertension. Now, if we going back to the regulation of oxygen homeostasis, so we do see the PHD2 is there, but PHD2 we know is controlled by oxygen levels, controlled by reactive oxygen species, and we also know PHD2 is controlled by some other signal pathways. For example, PHD1 and 3 can be induced some hormones. And also PHD1, 2, 3 can be regulated by the ubiquitous system as well. So far, majority of the regulation of PHD2 is on PHD1 and PHD3. So far, the only known regulation of PHD2 is hypoxia has a feedback effect to induce PHD2 messaging ion level. 2 is PHD2 can associate with OS9 as a oxygen sensing system to inhibit PHD2. So we don't really know much about the PHD2 in the literature. So that makes us interested into investigation of the molecular mechanism into PHD2 regulation, since we see PHD2 is dysregulated in pulmonary hypertension. So since we know PHD2 can be regulated at mRNA level or the activity level, so is there any other way PHD2 can be regulated? So we look at the another approach is more epigenetic approach, whether PHD2 can be epigenetically regulated. So we're looking at microRNA. MicroRNA are small non-encoding RNAs are known to suppress the targeted gene expression. So, and we know in our previous studies that microRNA 1792 is downregulated in pulmonary hypertension. So microRNA 1792 has 6 members, 17, 18A, 19A-B, 20A, and 92A. So some of them, they share similar seed sequence, some of them are unique. So we know PHD2 is up in smooth muscle cells from pulmonary hypertension patient, and the microRNA 1792 is down. So that gives us hint that PHD2 may be inhibited by microRNA 1792. So we're look back at the 3' UTR of PHD messaging RNA. So we are looking at PHD2 and PHD3. We do find PHD2 and PHD3 has a seed sequence for microRNA 28 and 17. That would suggest that these 2 proteins can be suppressed by microRNA 17 or 28. So we look at the protein levels. Yes, we're able to see microRNA 1728 to inhibit the PHD2 expression. And when we inhibit microRNA 1728, we're able to see induction of PHD2. And we look at the luciferase report. This basically trying to narrow down that seed sequence is responsible for the regulation of PHD2 by microRNA 1728. So these experiments we did, and we concluded that microRNA 1728 can inhibit PHD2 in pulmonary artery smooth muscle cells. Now, we want to see whether microRNA 1728 can inhibit PHD2 in smooth muscle cells. If they do, and they should affect the phenotype of these smooth muscle cells. So first, we want to see 1728 to inhibit the PHD2. Again, we use 1728 mimics in both normoxia hypoxic conditions, and we look at the PHD2. They are decreased, and the PHD1, PHD3, PHD1 no change, PHD3 is increased. Now, in hypoxic condition, we see 1720A inhibitor PHD2, 1720A inhibitor induced PHD2. So the system is working. Now we want to measure the function. So since we know that PHD2 is inhibiting HIF activity, so we will measure the HIF activity in the mice and the smooth muscle cells. So these are the mice with knockout of micron 1792. So if you don't have micron 1792, you have higher PHD levels. So the HIF activity will be down, the hematocrits will be decreased. So that's what we see here. A knockout 1792 will decrease hypoxia-induced hematocrit, hypoxia-induced red blood cell counts, or hemoglobin levels. So basically, in the cells, in the animals, when you take out 1792, you are seeing the induction of PHD2 and inhibition of HIF. And this is the mRNA level of HIF genes. Again, we are measuring the HIF activities. They're down. And this is a way of showing that micron 1792 can regulate HIF1 alpha, not HIF2 alpha. So that we are trying to link the micron 1792 to HIF and smooth muscle cell phenotype. Now, if we think the micron 1792 can regulate PHD2 to regulate HIF, and we know HIF PHD2 pathway is controlling cell phenotype. So next, we want to see whether PHD2 is playing a role in 1792 mediated cell phenotype. So in this case, we have a cell with inhibition of 1792A, and then we silence the PHD2, 1, 2, and 3. So we're trying to see that the BRDU incorporation in hypoxia conditions, normoxia conditions, and the ratio. So overall, when we silence PHD2, we are able to see the induction of BRD incorporation in normal conditions and in hypoxia conditions. So basically, this is saying silencing of PHD2 prevents inhibition of smooth muscle cell proliferation mediated by micron 1792 inhibitors. And so by showing these data, hopefully I can convince you that micron 1792 pathway can inhibit PHD2, mostly is through the micron 17 and 28. This inhibition of PHD2 will induce HIF1 alpha, induce smooth muscle cell proliferation, and pulmonary hypertension. So the data now I have been showing you has been published. However, in last year, there are 4 papers published along with us. So they all showing PHD2 is playing a role in pulmonary hypertension. So we are not alone in studying the mechanism into the hypoxia-induced pulmonary hypertension. Now, other 3 studies, they all focusing on the endothelial cells. So basically, our study is only one study focusing on the function of PHD2 in smooth muscle cells. But altogether, 4 papers show that PHD2 can control control vessel remodeling and pulmonary hypertension. One in smooth muscle cells that is controlling the HIF1 alpha, but in endothelial cells they are controlling through the HIF2 alpha pathway. So I have been showing some data in our system. HIF2 alpha is not in the picture. Now, in these cells, these studies, they also show the other downstream targets. They're more focusing on how does the PHD2 control HIF2 alpha, and that pathway, how does that recruiting parasites or controlling CXCL system to recruiting the progenitor cells to the vessels to increase the vessel remodeling. but none of these focusing on the upstream, what is regulating PhD2. So ours is only a study showing that microRNA 17 and 28 is inhibiting PhD2. But also I want to point out, because we are not alone, so in the research is always has a competition. So you have to do, whenever you want to study, probably somebody somewhere in the world is doing similar studies. So you want to do it quick, do it thorough. You want to do better, but sometimes you got to be careful when to submit it for publication. Actually, I can tell you, when we submit our papers, we were actually the other papers not out yet, but we got in trouble with reviewers because some reviewer point out or somebody also published that, but that time is not. But actually, 1 month later, that paper comes out. So that is something we unfortunately we have to live by when we do research. So time is important. Timing is important. Yes.
I disagree with that,
Okay.
because you really just, if you come up with the same findings, you're really supporting that research base. So I think even if you do come out with it at the same time, it doesn't matter. It's just more support that PhD2 actually works.
Well, it's
'Cause
true.
some people might do it and get a different result. You know how that can happen in research.
Oh, that's even
Yeah.
worse. Well, but for scientific point of view, yes, it's
Yeah.
true. You do different ways, different labs confirm the same finding. That's great.
Yeah.
But you know, for yeah,
Publisher cares, yeah.
for funding also it matters. So if somebody already got a similar result, got a funded, when you send out your grant proposal, it's unlikely you're gonna get a fund unless you have something new, right? But that's just I want to point out. But anyway, so whatever we find has been confirmed, or other studies have confirmed ours, or we confirm other people's studies, although in different way, different cell models. So now, so altogether, so hopefully I can convince you that PhD2 inhibits HIF1 to suppress pulmonary artery smooth muscle cell proliferation, and PhD2 is downregulated by microRNA 1720A in pulmonary artery smooth muscle cells, and activation of PhD2 is a potential therapeutic approach for PH. So with that, I'm going to conclude with my presentation, and these are people who did the experiment and the funding source. Thank you very much, and I'll be happy to answer any questions.
What we find clinically is that babies vary tremendously phenotypically with our pulmonary hypertension. Same baby, 1,200 grams, et cetera, et cetera, et cetera. So there are two issues here. One of them is, are they going to respond the same way to work on this particular pathway? And how are you going to sort that out? Or are you going to try and identify specific genomes that are sensitive? Because we really don't understand completely why, if we have 100 babies or 1,500 grams, that 2 of them are going to wind up with intractable pulmonary hypertension and the others aren't.
I think that's a good point. With everybody actually respond to the system differently. It depends on genetic background. So in our case, if you take out a PhD2 out of picture, these mice, they are more sensitive to hypoxia-induced pulmonary hypertension. So if a baby, they are missing the PhD2s, they will develop more severe pulmonary hypertension. They may not respond very well compared to other babies. So in that sense, yes, genetic background will affect how they respond. And also, sometimes it's about the threshold, how much they can take. Under certain stress conditions, some, even though they have a different genetic background, they don't develop any phenotypes right away. But under certain other stress conditions, it'll come out. So that is the perfect example to say how genetic background will affect the disease presentation or the treatment response. That's what we are doing now. That's what now precision medicine is in play now. A lot of resources have been going there. Now genetically, so far now we in pulmonary hypertension patient, so far there are no report on PhD2 genetic background change. But one thing it does come out, it's more regarding to the adaptation to high altitude. In people who live in high altitude, for example, the Tibet, and people who live in Andes in South America, they do show the difference in PhD2 and PhD1. Although people on Tibet plateau and people on Andes, their genetic background is very different. One is more the PhD2 HIF1 alpha pathway, another is more towards the PhD1 and HIF2 alpha pathway. So yes, in that sense, it will change. For example, some people, they go hiking, they go to high altitude, that where people develop the high altitude pulmonary edema. So if the background different, the way they respond to the climbing will be very different. So the treatment most likely will be a little bit different as well.
We're going to give all of our newborns coca leaves. Okay. Let them chew on it.
Okay.
Okay.
Yes.
Great talk. I have a question about the HIF2 alpha in the small muscle cell. So is there any function of HIF2 alpha in the small muscle cell? Because based on your system, it looks like PhD is regulated HIF1, but HIF2 was not. But I thought that there's HIF2 expression, so it's other functions. Even other functions not in this system.
That's good. So basically, there's a difference between HIF1 alpha and 2 alpha. So these two isoforms, HIF1 alpha is ubiquitous. Pretty much every single cell has expression. HIF2 alpha is more restricted. So the HIF2 alpha's name originally coming from called EPAS1. They think it's endothelial cell specific, but later on we found in other cell types, alveolar type 2 cells, smooth muscle cells also express HIF2 alphas. Now, your question regarding whether HIF2 alpha is functioning in smooth muscle, and the answer is yes, but not in this way. In our case, so mostly it's a PhD2 and HIF1 alpha, but we don't know if it's a PhD1 because in pulmonary hypertension patients, the PhD1 level changed, and PhD1 can regulate HIF2 alpha. Now, why I say in smooth muscle cells HIF2 alpha is functioning. So a few years back, actually, Dr. Arti Redheaven now is a faculty here. So when she was a fellow with me, we did a study on FOXM1 expression. So we found hypoxia can induce FOXM1 and then regulate smooth muscle cell proliferation and pulmonary hypertension. However, the hypoxia regulation FOXM1 is actually two ways. In continuous expression FOXM1, that is controlled by HIF1 alpha. But in hypoxia-induced FOXM1, that is controlled by HIF2 alpha. So in that sense, that's why I say HIF2 alpha is functioning, but it's in different pathway. In FOXM1, they can induce FOXM1 to promote cell proliferation. But in this case, PhD2 pathway, that is coming through HIF1 alpha. Yes.
They hit one pathway ends up being eventually down to the mTOR pathway. Has there been any work tried, and the mTOR pathway might be causing also contributing to the smooth muscle cell proliferation? Is there any work done trying to control the smooth muscle proliferation by knocking down the mTOR with rapamycin, like a lower dose, or maybe inhaled? I'm just trying to compare it.
Yes, actually, many data has been reported. I think the lab who does this lab work is the lady who used to be at Penn. Now she moved to Pittsburgh. Pittsburgh has a very large young research community. Yes, the mTOR pathway has been implicated in the smooth muscle cell proliferation, in the cell levels, in animal models, and that might actually, from some animal studies, can inhibit the proliferation. So that has been done. And a lot of work has been published.
Is there any diagnostic value for the Pediatric?
As for biomarkers,
that mean for early diagnosis?
We don't know yet because the PPD2 is the cytosolic protein mostly. If you want to do a biomarker assay, you may be able to do biopsies, but typically for biomarker research, you want to take some blood easy test for easy access. I'm not sure. So far, I don't see any reports on that. But in cancer field, there's some study going on now, but we are not there yet. So far, we don't know yet. That we need a lot more.
Thank you, that was so beautiful.
Oh, thank you very
Good
much.
job.